Today we are going to study about the microscopes and various techniques which are employed in microscopy. But before going into the detail of that, do you know what is the largest man-made structure? Yes, of course, it is the Great Wall of China. But do you know what is the smallest man-made structure? In 1990, using the very fine tip of a scanning tunneling microscope, a logo of IBM was created, which was just 17 atoms long and 6 atoms wide. It is the smallest architecture ever created. Today in this class we will be studying about these instruments which are called as microscopes and various techniques which are employed in the microscopy. Let's see a small video of our friends. Are they aliens? No. My dear friends, these tiny organisms are very well of our planet, but because they are so small in size, we could never learn about them. It is the invention of these microscopes because of which we are able to look at these tiny creatures. Let's learn about these techniques and the instruments which opened the windows for us to look into the tiny little world of these microorganisms. But first, let's define microscopy. What is microscopy? Microscopy is the technique of seeing or visualizing the objects which cannot be seen with unaided eyes. If one tries to define microscope, as its name suggests, micro means small and scope means to view. Hence, it is an instrument that allows us to look at small objects which can't be seen under normal circumstances. Microscopy is the technique because of which biology could reach up to its adulthood. You cannot think of modern discoveries made possible without the use of microscopes. Invention of microscopes in biology is like the invention of wheel in the human history. Different types of microscopes have been used to look at human cells, identify minerals, solve crimes, see how freezing affects food, study metals, and find the causes of crop diseases. They are an essential tool in medicine too. They are used to identify the causes of many deadly diseases like malaria and tuberculosis. They help to find out why a person or animal died. You cannot imagine the modern era of biology without the use of microscopes. So, are you ready to learn about the microscopes? Well, there are three branches of modern microscopy, that is, optical microscopy, electron microscopy, and scanning probe microscopy. A human eye can visualize structures up to 200 micrometer in size. For example, you can easily see a flea which has a size of about 1 centimeter. But anything smaller than 200 micrometer we are unable to see. For visualizing the structures smaller than 200 micrometer in size like amoeba or paramecium, we will have to use a compound microscope. However, a compound microscope also has got a limit. It is not able to visualize the structures less than 200 nanometer in size. For the structures smaller than that, like ribosomes or viruses, we will have to use electron microscopes or scanning probe microscope. Before entering into the science of microscopy, let's summarize the major historical events in the development of this science. In 1611, Kepler first suggested the way of making microscopes. In 1644, Leeuwenhoek reported the discovery of protozoa using his own constructed microscope. He also observed bacteria for the first time 9 years later. He is also considered as father of microbiology and microscopy. first functioning microscope using series of lenses was prepared by Robert Hooke in 1655. He observed the sections of cork and named them as cells. Because of this, he is also called as father of cell biology. Later, in 1833, Robert Brown described the cell nucleus for the first time by observing orchid cells under the microscope. From those times, our microscopes has evolved to a great extent, enabling us to see and visualize in far more depths and magnification. Now, before dwelling deep into the science of microscopy, let's first learn what a resolving power is. Resolving power is the capacity of microscope to distinguish or separate small objects that are kept close. The resolving power of the microscope depends on the wavelength and numerical aperture of the microscope. The lower the wavelength and higher the numerical aperture, the greater is the resolving power of the microscope. In this class, first we shall talk about the optical microscopy, then we will talk about the electron microscopy, and in the last session we will be talking about the scanning probe microscopy. Optical microscopy. As the name suggests, optical microscopy uses visible light for illuminating the sample. It simply passes visible light through or on the sample and then through a single or multiple lenses to allow a magnified view of the sample. The resulting image can be detected directly by the eye or imaged on a photographic plate or captured digitally. The most recent development is the digital microscope, which uses a CCD camera to focus on the exhibit of interest. The image is shown on a computer screen, so eyepieces are unnecessary. There are 6 basic types of optical microscopy. First, single lens. Second, compound light microscopy. Third, dark field microscopy. Fourth, phase contrast microscopy. Fifth, fluorescence microscopy. Sixth, confocal microscopy. Single lens microscopes are very simple, using only one lens. We also generally refer them as magnifying lenses. However, compound microscopes are pretty complicated, and we shall first learn about the compound microscope. Structure and function of a compound microscope. Now let's discuss different components of a compound microscope and their function. First, eyepiece. The lens the viewer looks through to the specimen are the eyepieces. The eyepieces usually contains a 10x or 15x power lens. Diopter adjustment. It is useful as a means to change focus on one eyepiece so as to correct for any difference in vision between your two eyes. Body tube, also called as head. The body tube connects the eyepiece to the objective lenses. Arm. The arm connects the body tube to the base of the microscope. Coarse adjustment screw. It brings the specimen into general focus. Fine adjustment screw. It fine-tunes the focus and increases the detail of the specimen. Nosepiece. It is a rotating turret that houses the objective lenses. The viewer spins the nosepiece to select different objective lenses. Objective lenses. It is one of the most important parts of a compound microscope, as they are the lenses closest to the specimen. A standard microscope has 3, 4 or 5 objective lenses that range in power from 4x to 100x. When focusing the microscope, be careful that the objective lens doesn't touch the slide. as it could break the slide and may destroy the specimen. Specimen or slide. The specimen is the object being examined. Most specimens are mounted on slides, flat rectangles of thin glass. The specimen is placed on the glass and a cover slip is placed over the specimen. This allows the slide to be easily inserted or removed from the microscope. Stage. The flat platform where the slide is placed. Stage clips. The metal clips that hold the slide in place. Stage height adjustment or stage control. These knobs move the stage left and right or up and down. Aperture. The hole in the middle of the stage that allows the light from the illuminator to reach the specimen. On-off switch. This switch on the base of the microscope turns the illuminator off and on. Illumination. It is the light source for a microscope. Older microscopes used mirrors to reflect light from an external source up through the bottom of the stage. However, most microscopes now use a low voltage bulb as an illuminator. Iris diaphragm. It adjusts the amount of light that reaches the specimen. Condenser. It gathers and focuses light from the illuminator onto the specimen which is viewed. Base. The base supports the microscope and it's where illuminator is located. The light is passed on from the illuminator, goes through the condenser lenses, passes through the specimen, goes into the objective lenses and to the eyepiece where we are able to see a magnified image. The magnification depends on the power of objective lenses and eyepieces. Compound microscopes can easily achieve the magnification up to 400x. However, when we move to the 1000x magnification, we observe that the image is getting little blurred. This is because of the fact that refractive index of glass and air are different. To overcome this problem, we use the oil immersion technique for higher magnification. Here, a drop of oil is placed on slide before viewing it under 100x objective lens. Oil used for this technique has same refractive index as that of the glass, and hence we can obtain higher numerical aperture and better resolution. This type of microscopy we have studied is also called as bright field microscopy because the field is bright while the specimen we are trying to observe is dark. Bright field microscopy has its limitation because of the fixed wavelength of light and a limit of numerical aperture. Apart from this, it does not offer a sufficient contrast, due to which we have to use stains to develop a contrast in the specimen. This generally involves fixing of cells which kills them and one cannot visualize live cells. I am sure you will be learning about various staining techniques in your practical classes. However, staining involves fixing of cells and fixation kills these cells, and we cannot visualize the live cells. To overcome this problem, a new technique has been developed which is called as the dark field microscopy. As you can see in the image, compared to bright field microscopy where the field is bright, dark field microscopy offers far more details. Let's look at this beautiful video of paramecium feeding. This is a type of dark field microscopy. As the name suggests, the field is dark and the specimen is bright, which is opposite to the bright field microscopy. Let's learn about this type of microscopy. Dark field microscopy. Dark field microscopy is an advanced form of microscopy where the lighting has been modified to reach from the sides into the specimen. Due to this specific arrangement, the only light reaching the objective lens is the light scattered by the specimen. Due to this, specimen appears bright on a dark background. As it employs higher numerical aperture, the magnification and the contrast achieved by the dark field microscopy is quite impressive compared to bright field microscopy. Apart from this, we do not need to strain the specimen to obtain the contrast, and hence it enables us to visualize live cells with detailed images. It is exceptionally good for observing motility of cell structures like cilia and flagella. One more type of microscopy which is good for observing the live cells is phase contrast microscopy. Let's learn about this phase contrast microscopy. Phase contrast microscopy. Phase contrast microscopy was invented by Dr. Fritz Zerkine in 1953. Phase contrast microscopy is an optical microscopy technique that converts phase shift in light passing through a transparent specimen to brightness changes in the image. Phase shifts themselves are invisible but become visible when shown as a brightness variations. Phase contrast microscopy is particularly important in biology as it reveals many cellular structures that are not visible with a simpler bright field microscope. Cellular structures were made visible to earlier microscopists by staining. This required additional preparation which killed the cells as we have already discussed. The phase contrast microscopy made it possible for the biologists to study live cells and how they proliferate through cell division. Now let's talk about another form of microscopy, that is fluorescence microscopy. But before we jump to the fluorescence microscopy, first let's understand what is fluorescence. Fluorescence is the property of some atoms and molecules to absorb light at a particular wavelength and to subsequently emit light of longer wavelength after a brief interval termed as the fluorescent lifetime. Using this property, we can explore the phenomenon of fluorescence. Fluorescence microscopy explores this fluorescence property of certain chemicals to develop far better resolution and contrast compared to light microscopy. The majority of the fluorescence microscope, especially those used in life sciences, are of epifluorescence design. In this microscope, light of the excitation wavelength is focused on the specimen through the objective lens. The fluorescence emitted by the specimen is focused to the detector by the same objective lens that is used for the excitation, which for greatest sensitivity will have a very high numerical aperture. Since most of the excitation light is transmitted through the specimen, only reflected excitatory light reaches the objective together with the emitted light, and the epifluorescence method therefore gives a better picture. The more advanced form of this fluorescence microscopy is confocal microscopy. It is the most recent and advanced form of microscopy. I am sure you will be studying about confocal microscopy in detail in your higher classes. Now let's move on to the next subtype of microscopy, that is electron microscopy. Optical microscopy uses photons as a light source. However, electron microscopy, as the name suggests, uses electrons as a source. Because they have a higher energy and longer wavelength, better magnification is achieved in electron microscopy. Now, before going into the detail of electron microscope, let's first define what is an electron microscope. An electron microscope is a microscope that uses electron beam to illuminate the sample and produce a magnified image of the sample. An electron microscope has greater resolving power than a light microscope and can reveal the structures of smaller objects because electrons have wavelengths about 100,000 times shorter than visible light photons. They can achieve the magnifications of as surprising as 10 million x, while compared to that, light microscopy can produce the images of about 2000x. So you can imagine the difference between the magnification of a light microscope and an electron microscope. The electron microscope uses electromagnetic lenses to control the electron beam and focus it to form an image. These electron optical lenses are analogous to the glass lenses of a light optical microscope. Electron microscopes are used to investigate the ultrastructure of a wide range of biological and inorganic specimens, including microorganisms, cells, large molecules, biopsy samples, metals, and crystals. Industrially, the electron microscopes are also used for quality analysis and failure analysis. There are two main types of electron microscopes. One is transmission electron microscope and second is scanning electron microscope. Transmission electron microscope. Transmission electron microscope is the original form of electron microscope. It uses the electron beam of high energy to produce the image. A high voltage electron beam to create an image. The electron beam is produced by an electron gun commonly fitted with a tungsten filament cathode as the electron source. The electron beam is accelerated by an anode with respect to cathode, focused by electrostatic and electromagnetic lenses and transmitted through the specimen, that is in part transparent to electrons and in part scatters them out of the beam. When it emerges from the specimen, the electron beam carries information about the structure of the specimen that is magnified by the objective lens systems of the microscope. The spatial variation in this information, or what we call as the image, may be viewed by projecting the magnified electron image onto a fluorescent viewing screen. Alternatively, the image can be photographically recorded by exposing a photographic film or plate directly to the electron beam or to a camera. The image detected by the CCD may be displayed on a monitor or a computer. Transmission electron microscopy played a key role in variety of biological discoveries. It is because of the transmission electron microscopy that we could learn about variety of organelles and their anatomy. Disadvantages of transmission electron microscopy. The major disadvantage of transmission electron microscopy is that it demands for ultra thin sections, and because of that, transmission electron microscopy is one of the costliest protocols in the microscopy. Now let's talk about the scanning electron microscopy. Unlike transmission electron microscopy, scanning electron microscopy is never carrying the entire image of the specimen. Instead, it is trying to collect the electrons which are scattered by the sample. The scanning electron microscopy produces images by probing the specimen with a focused electron beam that is scanned across a rectangular area of the specimen. When the electron beam interacts with the specimen, it loses energy by a variety of mechanisms. This lost energy is converted into alternative form such as heat and emission of low energy secondary electrons. These secondary electrons provide signals carrying information about the properties of the specimen surface such as its topography, and composition. Generally, the image resolution of a scanning electron microscope is poorer compared to a transmission electron microscope. However, because the same image relies on the surface properties rather than transmission, it is able to image bulk samples up to many centimeters in size, and it has a great depth of field, and so it can produce images that are good representations of the three-dimensional shape of the sample. Hence, on one side, transmission electron microscope gives us internal details. On the other side, the scanning electron microscope gives us the external morphology and external shape of the specimen. These two microscopes gave us the insight into many of the biological structures. However, a recent discovery of scanning probe microscope is even more advanced compared to electron microscopes. Scanning probe microscopy. Now let's study about this advanced form of microscopy, that is scanning probe microscopy. Scanning probe microscopy actually physically interacts with the specimen and tries to generate the information about the properties of the surface. An image of the surface is obtained by mechanically moving the probe in a raster scan of the specimen, line by line, and recording the probe's surface interaction as a function of position. SPM was founded with the invention of scanning tunneling microscope in 1981. The same microscope about which we discussed in starting, that how they created IBM logo using this scanning tunneling microscope. Scanning probe microscopes feel the changes in the surface of the sample. They use ultra sharp tips to measure the changes in the electric currents or electrostatic forces or magnetic forces coming from the surface. The magnification of scanning probe microscopy varies from technique to technique. However, some probes can achieve a magnification up to atomic level. There are two basic subtypes of scanning probe microscopy: atomic force microscopy and scanning tunneling microscopy. Now let's talk about the first subtype, that is atomic force microscopy. Atomic force microscopy works by interacting with the surface. The information is gathered by feeling the surface with a mechanical probe. The piezoelectric elements that facilitate tiny but accurate and precise movements on electronic commands that enable the very precise scanning. There are three basic types of atomic force microscopy: contact mode atomic force microscopy, non-contact mode atomic force microscopy, and tapping mode atomic force microscopy. I am sure you will be studying about these three in your higher classes. The scanning tunneling microscope. The scanning tunneling microscope is the ancestor of all the scanning probe techniques. Now let's study about this scanning tunneling microscope. It was invented in 1981 by Gerd Binnig and Heinrich Rohrer at IBM in Zurich. 5 years later, they were awarded the Nobel Prize in Physics for their invention. The STM was the first instrument to generate real space images of surfaces with atomic resolution. The smallest structure of the world we have talked about, the IBM logo, was created using this type of microscope. Summary. We started with optical microscopy and we learned how microscopes enable us to look into the tiny world. We learned about the anatomy and the functioning of a compound microscope. We also discussed its limitations, and then we talked about the dark field microscopy. We also explored the reasons why dark field microscopy offers better resolution compared to bright field microscopy. Later, we talked about phase contrast microscopy and how a far better resolution and contrast is achieved using phase contrast microscopes. We also discussed about what fluorescence is and how fluorescence microscopes are superior over conventional light microscopes. We also discussed about superior form of dark field microscopy, that is confocal microscopy. After completing the optical microscopy, we talked about electron microscopy and its two subtypes, that are transmission electron microscopy and scanning electron microscopy. Transmission electron microscopy gives internal structural details, while scanning electron microscopy gives topographical features of the specimen. In the last part, we developed idea about the most advanced form of microscopy, that is the scanning probe microscopy, which uses different types of probes to understand the surface features of the specimen.